Hello, and welcome to CRPS Contender, my complex regional pain syndrome education channel. Today we will be continuing with disrupted brain relations, another step down our journal journey discussing abnormal gray-white matter interactions in emotional and autonomic regions. This is part 2 of a very complex paper where we'll be discussing the real-world impacts of the test results that we did in part 1. Again, there is also a simplified version of this that has more description of the brain regions and images if you'd like to see where those regions are in the brain. This is a very complex paper, so if that's not your cup of tea, go ahead, check out the simpler version. This paper was published November 25, 2008 in Neuron by a group of researchers from Northwestern University and University of Toledo. Let's take from the abstract. Chronic complex regional pain syndrome is a debilitating pain condition accompanied by autonomic abnormalities. We investigated gray matter morphometry, which is the structural measures of the shape and volume of gray matter, and white matter anisotropy, which is measuring the connectivity of white matter by water molecule diffusion direction, in CRPS patients and matched controls. CRPS patients exhibited a disrupted relationship between white matter anisotropy and whole brain gray matter volume. Gray matter atrophy in a single cluster encompassing the right insula, right ventromedial prefrontal cortex, and right nucleus accumbens, as well as a decrease in fractional anisotropy in the left cingulum callosal bundle. Reorganization of white matter connectivity to these regions was characterized by branching pattern alterations as well as increased and decreased connectivity. While regional atrophy differentially related to pain intensity and duration, the strength of connectivity between specific atrophied regions related to anxiety. These abnormalities encompass emotional, autonomic, and pain perception regions, implying that they likely play a critical role in the global clinical picture of CRPS. Now we're going to hop right into the second part of this paper, starting in their discussion section. The main outcome of the current study is the observation that CRPS patients exhibit regional gray matter atrophy in a single cluster encompassing the right ventromedial prefrontal cortex, anterior insula, and nucleus accumbens. Quick refresher that ventromedial prefrontal cortex deals with a lot of social engagement, bringing together large concepts from different areas of the brain, abstract thought, moral judgments, executive decision making. The anterior insula does a lot of things, and it's not very well understood, but we know it deals with sensory motor processing, socio-emotional processing, and cognitive processing related to attention, salience, and speech. It is also the center of disgust, whether that is intestinal disgust due to eating rotten food or moral disgust. And then finally, the nucleus accumbens, which deals with reward and the interfacing from motivation to action. There's also a localized decrease in white matter anisotropy and changes in branching and connectivity of white matter tracks linked to these site-specific gray and white matter abnormalities. Unlike results from chronic back pain and fibromyalgia, whole brain gray matter and ventricular size were similar between CRPS and control subjects. Notably, the relationship between brain white matter skeletal fractal anisotropy and brain gray matter volume seems completely disrupted in CRPS, implying reorganization of white matter tracks in a manner that no longer conforms to the relationship seen in healthy subjects. Because this is a disruption at the whole brain level, it entails either diffuse disruption of gray to white matter relationships or multiple distinct abnormalities with compensatory remodeling of the brain in CRPS. Let's talk specifically about the anterior insula. Regional gray matter density comparison indicated atrophy within a single cluster for the whole group of CRPS, every single patient. The same brain region or portions of the same cluster exhibited atrophy even after subdividing the group by age or by laterality of CRPS pain. Hence, the atrophy spanning the anterior insula, ventromedial prefrontal cortex, and nucleus accumbens seems a robust result in CRPS and is right hemisphere dominant. Moreover, this atrophy was related to the two fundamental clinical characteristics of CRPS, duration and intensity, which impacted the density of this cluster above and beyond normal aging. When the cluster was subdivided into separate anatomical regions, the right anterior insula correlated with duration of CRPS pain. The insula is the brain structure most often observed activated in acute pain tasks. In CRPS patients, bilateral anterior insula activity correlates with ratings of touch-induced allodynia and pinprick hyperalgesia. Moreover, recent human brain imaging studies, consistent with older literature regarding the role of the insula as a viscerosensory cortex, highlight the role of the anterior insula in the representation of autonomic and visceral responses. Patients with pure autonomic failure due to peripheral disruption of autonomic responses exhibit reduced right anterior insula activity and atrophy in the right anterior insula. In healthy subjects, neural activity in the right anterior insula predicts subjects' accuracy in heartbeat detection, while local gray matter volume, at coordinates closely approximating the center of the cluster we observed atrophied in our CRPS patients, correlates with subjective ratings of visceral awareness. Furthermore, by comparing brain activity and autonomic responses in a fear conditioning task between healthy subjects and pure autonomic failure patients, colleagues conclude that the right anterior insula is involved in emotional representations, wherein feelings are the integration of both the mapping of internal arousal and conscious awareness of emotional stimuli. Given that CRPS patients are presumed to be in a constant negative emotional state and exhibit multiple signs of abnormal autonomic function, atrophy of the right anterior insula in CRPS corroborates the above studies and suggests that central anatomical abnormalities may explain fundamental symptoms of CRPS. Now let's shift our focus to the ventromedial prefrontal cortex and emotional decision-making. Atrophy in the right ventromedial prefrontal cortex was correlated with the interaction of duration and intensity of CRPS pain, which functionally segregates the atrophy in this region from the right anterior insula and suggests a more global impact or emotional load of CRPS on the ventromedial prefrontal cortex. Atrophy within this region was the author's primary hypothesis because CRPS patients perform poorly on emotional decision-making tasks, which has been shown to critically depend on an intact ventromedial prefrontal cortex. In fact, even when CRPS pain is transiently reduced, performance on emotional tasks does not improve, and CRPS patients do not show evidence of learning the task. In contrast, chronic back pain patients who exhibit atrophy in the thalamus and dorsolateral prefrontal cortex, although also abnormal on this task, exhibit clear signs of learning and improved performance over time. Emotional decision-making ability critically depends on the ability to evaluate options in terms of potential reward or punishment. Such decisions require proper capturing and evaluation of sensory cues, including bodily autonomic responses. Thus, it is not surprising that autonomic regulation and monitoring involve many of the same cortical regions implicated in emotional decision-making, especially the anterior cingulate cortex, the ventromedial prefrontal cortex, and the anterior insula. Therefore, differential atrophy of gray matter and abnormal connectivity of associated white matter tracts involving the anterior cingulate cortex, the ventromedial prefrontal cortex, and the anterior insula in CRPS, in contrast to the atrophy of the dorsolateral prefrontal cortex found in chronic back patients, must underlie the CRPS patients' differential responses on emotional decision-making, especially given the fact that CRPS is associated with autonomic abnormalities and chronic back pain is not. Neurons within the ventromedial prefrontal cortex encode the emotional value of sensory stimuli. Moreover, patients with ventromedial prefrontal cortex lesions exhibit diminished emotional responses and social emotions, as well as poorly regulated anger and a low frustration tolerance. The authors also demonstrated that the strength of white matter connectivity between the ventromedial prefrontal cortex and the nucleus accumbens was related to the heightened anxiety generally seen in such patients. I'm going to shift focus a bit to voxel-based comparisons of fractal anisotropy, which showed a single cluster with decreased anisotropy in the left callosal bundle, decreased parallel diffusivity, and increased perpendicular diffusivity. The decreased parallel indicates axon damage, and the increased perpendicular indicates demyelinization. This can be interpreted as an injured white matter bundle. Connectivity from this cluster in CRPS was lower for long distances, and local branching patterns were less space-filling, creating less connectivity. Both results are consistent and complementary to the notion of loss of axons, injury, or both. Therefore, it appears to be an independent brain injury observed in CRPS, unrelated to gray matter atrophy. Decreased fractal anisotropy in a region closely approximating the area observed here has been described in obsessive-compulsive disorder, as well as other mental health conditions. The injured bundle seems to affect connectivity of the anterior cingulate cortex as well as the supplementary motor area, a region repeatedly implicated in acute pain perception, especially the affective or emotional component. Now let's interrelate gray matter atrophy with white matter reorganization. The decreased gray matter density in CRPS may be due to various neurobiological causes, such as atrophy secondary to excitotoxicity and/or exposure to inflammation-related agents such as cytokines, decreased cell size, synaptic pruning, decreased number of glia with little or no neuronal death, or genetic predisposition. Examining white matter properties for gray matter regions showing atrophy enabled the authors to exclude some of those mechanisms. Thus, in addition to atrophy, white matter properties also suggest rewiring with evidence for synaptic pruning and target-specific rewiring. While the causal relationship between these changes and CRPS onset remains unclear, the correlation between brain atrophy and CRPS duration suggests that at least some of these gray matter changes are a consequence of living with CRPS long term. Now let's discuss some technical issues that they had with this paper. Even though both methods are validated and reproducible within and across subjects, the sensitivity and specificity of DTI remains less explored. Because both approaches use whole brain repeat measures and corrected comparisons, they invariably only identify the most robust abnormalities. And it is quite likely that the single fiber-based approach used here for probabilistic tractography missed some tracks, especially in the decreased fractal anisotropy area where the smaller cingulum and larger callosal pathways cross over each other. As a result, the authors may have underestimated the contribution of the former. In conclusion, the authors provide evidence indicating that the patient with CRPS has multiple pathological changes of the brain. They observe global disorganization of the relationship between gray and white matter in CRPS subjects. Regional gray matter atrophy seems limited to brain regions that can relate to these patients' deficits in emotional decision-making and abnormal sympathetic outflow. Regional white matter anisotropy was observed in a bundle in the hemisphere on the opposite side to the gray matter atrophy, where long-distance connections and branching patterns were reduced. The interrelationship between gray matter atrophy and white matter connectivity provide evidence for both decreased long-distance connectivity and regional increases and decreases in connectivity and changes in branching patterns. These results suggest that the abnormal anatomy of the CRPS brain may underline many of the autonomic, cognitive, and pain abnormalities seen in this pernicious syndrome. If you would like to help my channel engage more with the algorithm, please like, comment, or subscribe. If you'd like to contribute to my living expenses, my Patreon is CRPS Contender. Thank you to Emily Malcontent for being a loyal patron. If you'd like to see the paper, notes, or slides used, they are linked in the description. Thank you so much for watching. I hope you learned something, and I hope to see you next time.